Hello, everybody. This is Miss Peachy from your WCA biology class. And what we are looking at today is Unit 8, Lesson 3. We are still talking about genetic variation. So we are looking at genetic variation from errors this time. Your vocab for this unit include DNA, DNA polymerase, inheritable genetic variations, mutations, replication, viable errors, cell theory, substitution mutation, insertion mutation, frame shift mutation, point deletion, epigenetic changes, epigenome, methylation, undermethylation, and overmethylation. So quite an extensive list of vocab terms that you have here. So basically what this unit is, or what this lesson is trying to show you is that there are other ways other than just the standard meiosis to introduce genetic variation into organisms. So normally during meiosis, you have crossing over that occurs and you have independent assortment, which will normally lead to variation in any population, right? That's why you don't look exactly like your brothers and sisters, because you have a 1 in 8 million chance of having the exact same genome, and then that's just on your mom, but then their dad also has a 1 in 8 million chance, right, of having the exact same genome. So that, like, the chances of you and your brothers or sisters looking identical are like slim to none, unless you are come from the exact same, original cell, like with twins. So where do other variations come from? And the answer is that variation can also be introduced through mistakes, through errors. I don't even like calling them errors and mistakes, because sometimes these are really good things. So they're like good mistakes. And sometimes they can lead to like bad, lethal mistakes as well. So mutations, by definition, are any changes made to the DNA sequence. These can be favorable, they can be neutral, or they can be unfavorable. Kind of depends upon the outcome, really. And if this benefits the organism, does have any effect on the organism, like pretty much you don't even know it's there, or is negatively affecting the organism. Any kind of mutation that is inheritable, however, needs to be able to be passed on from parent to offspring, has to then occur in such a way that it affects their eggs and sperm. The only way you are going to be able to pass a trait onto your offspring is if that error occurs in those cells. So either it has to occur and affect your eggs and sperm, or it has to occur developmentally while all that is forming in your body, right? So then it becomes kind of a part of your egg and sperm once you become a fully fledged human. Um, so it talks a little bit about here some of the vocab that we've already revisited before. It kind of reminds us of the structure of DNA. That DNA is this double-stranded helix. It has a sugar phosphate backbone, and the two strands of DNA are joined together by these nitrogenous bases in DNA, that is adenine, thymine, cytosine, and guanine. That DNA, I'm skipping ahead a little bit here, is replicated or copied. In fact, DNA is copied either if you do mitosis or meiosis. So most errors, mutations, are going to occur during cell division, because that's, this is the largest chance of having a mutation. And cell division is mitosis or meiosis. And errors that are inheritable occur during meiosis in general. Again, darn you, meiosis. You've introduced so much genetic variation into the population, both intentionally and unintentionally. But this replication process begins when that double-stranded DNA is unzipped and unwound, and then it is copied so that the old strand of DNA serves as a template to make a new strand of DNA. The copying is done by an enzyme called DNA polymerase, and DNA polymerase does a bunch of different things. It not only copies, but it also proofreads a bunch of stuff as well. But it is an enzyme that plays a very critical role in placing those nucleotides, or those nitrogenous bases, which aren't exactly the same thing, but for all intents and purposes this time we're kind of calling them the same thing, in the correct order. All those A, T's, C's, and G's, right? Now remember, during the process of replication, you're kind of making new chromosomes to be able to go on into that cell division process, right? The other thing we see here is during when proteins are made, so during protein synthesis. So during protein synthesis, we have amino acids that are produced, and they're linked together to make proteins. Each amino acid is determined by a 3-letter sequence on the messenger RNA. So that messenger RNA is produced during transcription. It leaves the nucleus and finds a ribosome to attach to and is read 3 bases at a time. Each 3-base sequence we call a codon. So one codon at a time is read. And each codon codes, that's where the word comes from, for an amino acid. So UUU will code for a different amino acid. It says here the UUG represents the amino acid leucine, right? So that code is very important. So when there's mistakes in the code, that can lead to mistakes in the amino acids, and those can lead to mistakes in the proteins that are made as well. So let's kind of get into some of these errors, shall we? So our first error type of mutation is called a substitution mutation. And a substitution mutation is where the wrong base is substituted. So nucleotide bases are represented by the letters A, T, C, and G, with A pairing up with C and C pairing up with G. If any of the bases are mistakenly exchanged for another, the mutation is called a substitution mutation. So here we have the original strand ACC, GGC, CGA, ACA, and GGG, right? And here we have the mutated strand. So everything looks similar. Uh-oh, we got a problem right here. Before we had CGC, and now we have CCC. Before, our arginine was produced, and now proline is being produced. So we're producing the wrong amino acid. So that occurs during a substitution error. When can this happen? This particular process probably happened during DNA replication. So when that DNA polymerase was kind of putting all those correct bases all in a row, it screwed up and put a wrong base in there. And so we have a wrongly replicated strand of DNA, which in turn leads to an incorrect amino acid that is being produced. Will that affect the protein? Don't know for sure. If it does, it's a negative error. If it's a neutral, then it won't have any, any kind of result from that. We'll just call it a neutral error. And otherwise, it could be positively affecting, which is rare, but it does happen. Okay, in one such example they're giving here of a substitution error, they're giving something called sickle cell anemia. So for example, if G is added instead of A, the G will pair up with T. This could create a codon that changes the amino acid being produced. This in turn could cause a small change in the protein that is created. Sickle cell anemia is a disorder caused by a substitution mutation, in which a single amino acid is altered in the beta hemoglobin gene located in red blood cells. And humans that have sickle cell anemia, they don't have the proper shape for their blood cells, so they don't carry oxygen very efficiently, and so they become very, very sick because you need oxygen for what? What is the process, you guys? Respiration, right? You need oxygen for respiration. So if your body can't officially do cellular respiration, you're going to be tired all the time because you can't get enough energy. You're not producing enough ATP. You won't be growing properly, and it can cause things that it can even cause organ failure. It can be really, really catastrophic. So silent mutations or neutral mutations are harmless because the substitution creates a codon that still produces the same proteins. And if the substitution creates a stop codon, the copied strand will produce an incomplete protein. This type of protein will not function properly, and sometimes it's called a nonsense mutation. So there's multiple scenarios to that, but the bottom line is you substitute the wrong thing in, it could have a negative effect, could have no effect, very possibly it could have a positive effect. It's rarely possible, but it does happen. Insertion mutation is just instead of, you know, um, putting the wrong thing in, you're putting something extra in there. Okay. So an extra base is put in. And this can be kind of more catastrophic to some degree because it ends up being something called a frame shift mutation. And this kind of depends on how many bases are inserted. So let's say you just insert one additional base. Well, then everything moves over one. Remember, they're read 3 codons at a time. So if I stick something in the middle, all of a sudden we're reading it, these 3, and then, I can't do this. I'm doing it backwards in the mirror. So these 3 are read first, and then these 3 are read, and then that one's left over. When normally, if I hadn't inserted that, it would just be these 3 are read, and then these 3 are read. Do you know what I'm saying? So if we put something in the middle, all of a sudden we read 3, 3, and we leave 1 out. I'm pretty sure I screwed that up because I'm trying to look at myself in there compared to myself in person. So normally we would have just these 3, right? So we read these and then read these. But if we stick something in the middle, now we're reading these and then reading these, and we're not getting to that guy. Does that make sense? And so it also gets messed up. It's not like you're just, reading them out of order. It's like the letters are now out of order. So I think we have an example of that too. If the original was ATC, GAG, ACC, those are your 3 codons. But if all of a sudden you stick something in there, in this case, now we have ATC. We stuck an A in there, right? That dark A. AGA, GAC. So now this amino acid is wrong. This amino acid is wrong. Every other one will be wrong because you inserted 1 base. If the insertion is a full 3 bases, it won't matter because you'll just kind of skip over that. It probably will, it could still mess things up, but the likelihood is a lot less. Then we have deletion mutations, and you guessed it, they're taking one of the letters away, at least one, sometimes more than one, right? So if you delete one of the letters here, we have ATC, GAG, ACC. Now we have ATC, we're missing the G. And again, everything shifts. So it becomes another type of a frame shift mutation there because you're shifting the frame. Unless you're deleting all 3, that could have some really catastrophic effects. So a point deletion is when 1 base is missing. Cystic fibrosis is an example of a disease that's a result of a point deletion on the CFTR gene that affects the proteins that move salt and water in and out of cells, creating mucus buildup in different areas of the body. So that's an example. All right, so inheriting errors. Many replication errors can contribute to inheritable genetic variations because the changes in the genetic material are viable. So viable means you can still live, basically. So if the mutation is such that you still can be alive and reproduce even with that mutation, then you can pass that mutation off to your offspring. Oftentimes, mutations that occur early in development result in death of the embryo, and they're never passed on to the next generation because they're not viable. But those that are viable become viable heirs because they can be passed on to the next generation. However, even these viable heirs will oftentimes lead to severe health disorders, shortened lifespans, or even sterile offspring. Okay, and I believe there is one more section here. Talk about epigenetics or epigenetic changes, right? So basically, this is talking about how there are things that affect replication of DNA. So you have these proteins and other chemical compounds that attach to DNA, and they regulate whether or not that DNA is replicated. That either can turn some genes on, which then you replicate them, or I shouldn't say replicate, those are read and proteins are transcribed and translated from there, or it turns it off and protein synthesis doesn't happen, right? And so the epigenome consists of proteins and chemical compounds that attach to DNA and can turn certain genes on or off, affecting the production of proteins in cells. Epigenetic changes occur when the expression of the gene is altered and not the actual DNA sequence. So instead of like mutating the DNA, it's altering what is actually being transcribed and translated from the DNA. It's affecting the reading of the DNA. It's either causing the reading not to happen too much, or to happen too little. Okay? And so it talks about some processes here. An example is a process called methylation, which involves attaching methyl groups consisting of one carbon atom and three hydrogen atoms to segments of DNA. You don't need to know that part. When it attaches the methyl group to the DNA, the gene is turned off and protein production stops. Excuse me. This action can lead to abnormal gene activity or inactivity and cause genetic disorders for people with high risks of some disease. Undermethylation is when there are too few methyl groups to regulate gene expression. Having too few methyl groups can cause symptoms such as obsessive compulsive disorder, increased fear, frequent headaches, or poor concentration. Interesting. Studies are being conducted to understand the connection between undermethylation and severe neurological disorders such as depression, anxiety, autism, schizophrenia, and Alzheimer's disease. So there's too much gene expression happening here. Overmethylation is when there would be too little gene expression. There are too many methyl groups trying to regulate gene expression. Sometimes symptoms for people who have overmethylation include panic attacks, sleep disorder, body aches, food or chemical sensitivities, and paranoia. In similar studies of undermethylation, overmethylation has been linked to neurological disorders such as depression, anxiety, and schizophrenia. Roughly 45% of schizophrenic diagnoses show the patient also has an excess of methyl groups. Very interesting. So these methyl groups, they regulate, meaning that they kind of slow down protein synthesis. And so it's just one way in which errors can be introduced, either by doing it too little, or too much, essentially, than you need to. And it can cause errors. These are, again, viable errors because they can exist and still be then that the person with that error can still reproduce and pass on that error to their offspring. All right, so that is the end of Unit 8, Lesson 3 on genetic variation from errors. Wow, a lot of vowels there. Stick around. I'll see you in the next video.